Thanks for coming out, guys. So, life is a cycle of birth, growth, and death. And in order for life to continue, the genes and that genetic information has to be passed along somehow. And in order to do this, there's a process called replication. And that takes the DNA, your deoxyribonucleic acid, and copies it onto a new strand. Now, as a biology major, I've taken a bunch of courses regarding biology, obviously, and I've learned a lot about DNA replication year after year. And I'm going to tell you a little bit about how the process works and where mistakes can happen. And I guess I'll get started with the process. So DNA replication is a semi-conservative process, which means there is one parent strand and one daughter strand, two parent strands and two daughter strands at the end, but one parent strand with one daughter strand. And so you have one original strand with one new strand. That's what it means by semi-conservative. And this, most of the information I got from the textbook Genetics, A Conceptual Approach by Pierce. And it's a very straightforward process, but there's a bunch of things that are involved when you're actually talking about DNA replication. So the first thing that needs to happen with DNA replication is you need a helicase to bind to the site of origin, and the helicase binds to the site of origin and it unwinds the DNA. It breaks the hydrogen bonds, which are represented right here, between the nucleic acids on the different strands. So when the helicase is bound, it's going to start unwinding, but in the process of unwinding this direction, it's also unwinding on the other side because it's bidirectional. And as it unwinds, it makes the strands very unstable. So there are single-stranded binding proteins along the strand to stabilize the single strands because DNA wants to be a double-stranded molecule. In front of the helicase, there is an enzyme called topoisomerase, which goes to reduce the torsion created by the unwinding of the DNA strands. And DNA is, So there is another enzyme called RNA polymerase, which is similar to DNA polymerase. It can add on nucleotides. However, RNA polymerase unwinds and does both processes of a helicase and a DNA polymerase. And after the strands are unwound, you have a DNA polymerase coming in and binding on to a primer created by primase. So the DNA polymerase cannot bind to an open strand, and it has to have something bound on there. So that's what the RNA primase does. So once the DNA polymerase is bound, it's able to add on nucleotides consistently and continuously as long as it's moving in the 5' to 3' direction. Now there's a lagging and a leading strand. The leading strand is continuous in its replication, while the lagging strand is discontinuous because the lagging strand is moving this direction away from the fork. So you can only move so far before another piece opens up. You only have so much open up at one time. And once the lagging strand is being completed, it needs to be brought back together. It needs to be connected, those pieces. They're called Okazaki fragments, and they're connected by an enzyme called ligase and another DNA polymerase. So once you have those two strands completed, you have your two separate newly replicated strands. However, the process isn't as simple as that there is mutations, obviously, that happen. There are three main types of mutations. There is substitution, insertion, and deletion. Substitution is the least detrimental type of mutation to occur, and there are three different types of substitution. There's nonsense, which changes the amino acids so that it creates a stop codon when you're creating proteins. And there's the missense mutation, which changes the last codon to code for a different amino acid. And then there's finally the silent mutation, which changes the codon, but it still codes for the same amino acid, which means you still get the same protein. And then insertions and deletions cause frame shifts. So when a couple nucleotides, or even just one nucleotide is added, it changes every other codon created all the way down the line, and actually normally causes cell death because a protein can't be made. The same protein that is supposed to be made is not being made. Now, those types of mutations can all be fixed. There is the DNA polymerase, which helps connect the Okazaki fragments that I was talking about. That can go in and switch out the incorrect nucleotides in something like a substitution. And then there is the endonuclease, which cuts out that piece of incorrect information, and then the DNA polymerase comes back in and it corrects everything and connects all the pieces again. So that's a little bit of a rundown on DNA replication and the problems associated with it. It's a very straightforward process, but it does get confusing when you add in mutations and switching out nucleotides for the correct nucleotide. But it is a very important process because it is how life continues, and it's what we need in order as a species to continue, as other species continue. Thanks for coming out.